Hello, it's great to be with you all. My name is Dr. Craig McDonald. I'm a professor and chair of physical medicine and rehabilitation and director of the neuromuscular disease clinics at UC Davis Health in Sacramento, California. I'm also the study chair for the CINRG Duchenne Natural History Study, where we collected natural history data for 10 years. And I'd like to share with you today our upcoming approach to a new launch of an expanded Duchenne natural history study. I'm going to be discussing that new study with you and also give you some thoughts about what we've accomplished with the previous Duchenne natural history study. Just as a matter of disclosures today, I have done consulting work on Duchenne muscular dystrophy clinical trials for a number of pharmaceutical companies, and I've also received research funding for the conduct of clinical trials from a number of these pharmaceutical companies as well that are doing clinical trials in the Duchenne muscular dystrophy space. There's really nothing in my talk today pertaining to any product that is being developed commercially for Duchenne muscular dystrophy. I'm really going to be focusing on concepts regarding our expanded natural history study. And also, I'd like to provide some perspectives on the challenges of conducting clinical trials in the era of the COVID pandemic. So just to give you some background and history, the original CINRG or Cooperative International Neuromuscular Research Group Duchenne Natural History Study was conducted in over 20 centers internationally, again following patients, over 400 Duchenne muscular dystrophy patients for periods of up to 10 years. This has been an extremely productive effort. For those of you that perhaps might have questioned the value of natural history studies, this work has actually resulted in 28 peer-reviewed publications. This effort, in collaboration with multiple investigators worldwide, has helped define genetic polymorphisms which impact the nature of the disease progression with Duchenne muscular dystrophy. We've helped identify blood biomarkers, and there have been actually more than 25 cases where industry has actually used our CINRG Duchenne natural history study data to help plan future clinical trials. The data has also proved to be critical in a number of regulatory evaluations conducted both by the FDA and the European Medicines Agency. A couple of examples recently is the case of vitilarsen, the exon 53 skipping agent, which is being studied by NS Pharma, as well as adebenone, where they have a planned confirmatory trial referred to as the MELOS study as part of their European submission, which will actually use the new CINRG expanded natural history study for their comparisons. And I think increasingly, the regulatory authorities have recognized the value of some of these case control comparisons that have been conducted. And also, we're also realizing the natural history data can actually be used to enrich a smaller placebo group in the conduct of clinical trials. I mentioned the number of publications from the CINRG Duchenne Natural History Study. This is focused on topic areas such as the nature of Duchenne muscular dystrophy disease progression and some modeling efforts focusing on issues of strength and function, the benefits of long-term steroid therapy with regard to preservation of function, both in terms of ambulatory function and upper limb function, long-term benefits of steroids and natural history data concerning pulmonary function, and then natural history of cardiac function and cardiac care utilization, biomarkers of disease progression and steroid response, and then some validation of the clinical meaningfulness of some of the clinical endpoints used in Duchenne muscular dystrophy clinical trials, using anchoring-based techniques, comparing those clinical endpoints with person-reported outcome measures, or PROs. The data has been used widely by academic physicians, industry sponsors, and regulatory agencies, both the FDA and the EMA, to help with clinical trial planning and also to provide some context for the evaluation of clinical trial results as well. Just to give you some recent examples of the impact of some of these publications, I mentioned 28 publications. There's been over 800 citations in peer-reviewed journals relating to these 28 publications, on average 30 citations per paper, using the newly available NIH eyesight statistics. In addition, this research has had impact, again, using the NIH eyesight statistics. The relative citation ratio numerical value has been 3.97, which is actually quite remarkable. Has been excellent. The mean citation ratio for NIH-funded research is a 1.0, which would place that at the 50th percentile. The Duchenne natural history study papers actually have a citation ratio which scores at the 92nd percentile. So really quite impactful in relation to NIH-funded research conducted to date. So let's address this important question of how has the COVID-19 coronavirus pandemic impacted the conduct of Duchenne muscular dystrophy clinical trials. I think we're all keenly aware of the impact that this has had on our clinical management, our clinical care, in addition to the conduct of clinical trials. So this impact obviously has varied by state. There's been some temporal issues that has, so the impacts have changed state by state over the months of the COVID-19 pandemic. It's also, even within a state, it is also varied by institutional clinical research policies, which have impacted the conduct of clinical trials, the degree to which patients can be brought into the academic medical centers for clinical trial visits. Also, on an emerging basis, both in-person and video visits have been completed to allow us to continue to collect valuable data conducted during clinical trials. This certainly, the conduct of clinical trials is certainly realistic, even in the face of a pandemic. Just to give you an example of the burden and impact that this has had on the clinical research teams for the past 4 months, to give you an example, at UC Davis, we have actually executed over 40 IRB protocol amendments just on Duchenne muscular dystrophy trials, and these amendments being COVID-19 related amendments. Despite that, we at UC Davis, for example, have been able to successfully conduct 98% of our clinical trial visits during the COVID-19 pandemic. But I think moving forward into the new future, I think we need to think creatively with regard to how data is going to be collected in Duchenne muscular dystrophy trials. And also, COVID has created challenges for the conduct of natural history studies. These studies are oftentimes, since they're not interventional studies, these sometimes are deprioritized in terms of the ability to bring patients in for natural history study visits. I think as a emerging concept, I think these natural history study visits can be conducted as long as they're consistent with the conduct of standard of care, standard of care with regard to clinical follow-up and clinical visits. I think there's also been, I think, some emerging outcome measures which are being evaluated, which are perhaps home-based measures or the increasing use of wearable technologies, which is really quite exciting. There are some patient-oriented outcome measures which allow data to be collected in the home environment, which are really quite exciting. But I think there's an important concept and issue to keep in mind is that now is really probably not the time in our clinical trials to implement completely new primary outcome measures for these DMD trials. First of all, many of these novel outcome measures that are being collected in the home environment lack long-term natural history data. They lack prognostic data concerning prognostic relevance with regard to the relationship of 1 and 2 year changes to long-term clinically meaningful outcomes, and just the definition of clinically meaningful treatment effects is largely unknown with some of these emerging candidate outcome measures. The clinically meaningful treatment effects are really critical to a regulatory approval. So I think part of our emphasis in our new expanded Duchenne natural history study, as well as our previous study, has been to focus on some of the well-established clinical endpoints and outcome measures that have been utilized in Duchenne muscular dystrophy natural history studies, as well as Duchenne muscular dystrophy clinical trials. So I'd like to just briefly go over our new CINRG expanded natural history study. This is the eDNHS study, expanded Duchenne natural history study. This study has been approved at our UC Davis site. We're ready to launch that. We have 3 industry sponsors for this study. We're really excited about launching this new expanded Duchenne natural history study. In terms of number of participants, our goal is to ultimately continuously enroll and follow up 500 participants at any given time. So participants who are lost due to attrition or perhaps transition onto a clinical therapeutic trial or through morbidity or death will be replaced based on their age of study discontinuation. So there'll be a robust natural history sample size moving forward. The eligibility criteria are actually very similar to the previous Duchenne natural history study, the previous CINRG Duchenne natural history study, with the exception that we are actually going all the way down to birth onwards. The previous natural history study enrolled patients who were 2 years of age and above. The patients will have centrally confirmed by a central genetic counselor diagnosis of Duchenne dystrophy based on molecular genetic criteria or muscle biopsy with dystrophin immunofluorescence or Western blot showing complete dystrophin deficiency, and also a clinical picture consistent with Duchenne muscular dystrophy. So again, the mutation subtypes that we're focusing on is where we have deletion duplications or point mutations, which produce a reading frame predicted as out of frame in patients with a clinical picture consistent with Duchenne muscular dystrophy. So the criteria are really quite similar to the previous study that was done. The number of trial sites, we're targeting approximately 20 U.S., non-U.S. Sites. Participants will be seen every 12 weeks, which is very similar to many clinical trials which are conducted in this day and age with Duchenne dystrophy. The first year will go through week 48, and then the patients will be followed every 20 weeks, plus or minus 2 weeks thereafter until week 252 or year 5. The following assessments will be completed: vital signs, a review of ambulatory status and steroid medications or other disease-modifying medications, a review of any nutritional supplements, cardiac and pulmonary medications, a review of systems, including key systems, and then a number of clinical evaluator assessments will be obtained as well. Standard anthropometric assessments, goniometry, evaluating for contractures, the key time function tests, including stand from supine, standing from a chair, has been added, 10-meter walk run time, the 100-meter run walk test, and the 4-stair climb. Functional tests, including the Brook and Vigno scale, as well as the pull entry items, the 6-minute walk test, the North Star ambulatory assessment, the EK, and performance of upper limb version 2.0. Pulmonary testing will be also obtained, including FVC, FEV1, the peak expiratory flow rate, the maximal inspiratory pressure, maximal expiratory pressure, and the inspiratory flow reserve. And then some quantitative strength testing will be obtained unilaterally, as well as grip testing. And then some sites will actually be studying community activity monitoring as well moving forward. A number of PRO assessments will be obtained, including the PODCI questionnaire, which has been, we have a wealth of data, over 10 years of data. Many of the PROMIS items have been included in the NeuroQual, but there's also the pediatric PROMIS instruments, the PROMIS fatigue questionnaires, the upper limb PROM. There's a new dysphagia questionnaire called the EAT-10, the Duchenne muscular dystrophy lifespan mobility scale that has been validated by Dr. Eric Henriksen, the EQ-5D, and then some parental questionnaires concerning parent-caregiver work activity, work productivity, and activity impairment. These have been adapted for neuromuscular disease and Duchenne patients. Again, a patient or parent-reported review of systems. There's biomarker sample collection and evaluation of adverse events. In the very young patients, again, very similar protocol, with the exception that the North Star ambulatory assessment will actually be obtained as we're able to with these patients, as well as the Bailey-3 scales of development. If these patients are able to be equipped with a wearable device, community activity monitoring will be obtained, and then the PRO assessments will focus more on the proxy and parent reported outcomes. And then biomarker sample collections will be obtained as well. I want to just comment on some novel assessments that are going to be part of the CINRG expanded Duchenne natural history study. We are currently in the process of validating the Nuvo Air home-based spirometry system, which gives you values for FVC, peak expiratory flow rate, and FEV1. This has actually been used in industry-sponsored trials of COPD and asthma We're actually quite excited about this. So far, the data looks very good in Duchenne muscular dystrophy patients. Also, home-based transcutaneous CO2 monitoring will be obtained in a subset of patients to define the actual onset and occurrence of nocturnal hypoventilation. Again, many of the Duchenne care considerations that give certain FVC milestones for when nocturnal ventilation should be initiated were largely due to consensus recommendations from experts, but were not necessarily data-driven. And the EMA has pushed back on this concept of using particular milestones of FVC values as a clinically meaningful endpoint transition to a milestone value of FVC. And the EMA has actually encouraged the actual acquisition of data concerning respiratory failure and CO2 retention. And so we'll actually be obtaining some home-based transcutaneous CO2 measures. We're also in the process of doing some validation work on a video-based North Star, as well as some validation work on a video-based performance of upper limb assessment. And then finally, video-based time function tests conducted in the home environment. I think these are all going to be important because in the setting of infectious disease considerations and perhaps surges in the COVID-19 pandemic, I think it's not always guaranteed that patients are going to be able to come into the clinic environment, particularly for natural history study assessments, even for clinical trial assessments. So we're really using this natural history study as a test bed to evaluate some of these novel home-based assessments. And then we're also going to be studying, as I had mentioned, some novel PRO assessments in the Duchenne population as well. I want to just give you, to finish by giving you a brief update on some exciting work we've been doing in collaboration with Santhera, using the CINRG natural history data to define a minimal clinically important difference for respiratory function in Duchenne muscular dystrophy as measured by spirometry or pulmonary function tests. And the question is really how to derive the minimal clinically important difference for a pulmonary function measure, measure of respiratory function. I think that some of the statistical distribution properties that have been employed are not necessarily clinically driven, and I think oftentimes the regulatory authorities would like to see some sort of anchor-based approach or prognostic value with regard to the onset of a clinically meaningful event. And in this case, we have focused on the actual onset of mechanical ventilatory assistance as collected in the Duchenne natural history study, which was conducted prior to the recent care considerations where mechanical ventilation has been recommended for patients reaching an FVC of 50%. We actually collected data on when mechanical ventilation was actually initiated in a large cohort of Duchenne patients studied prospectively for up to 10 years. So here's an example of the linear decline that we see with percent predicted forced vital capacity and peak expiratory flow rate. The interventional studies have usually assessed the rate of respiratory function decline during a fairly short window of, say, 1 year in duration or up to 18 months. And the question is, when we look at the short-term treatment effects, perhaps the FVC was the rate of decline in forced vital capacity percent predicted value was decreased by, say, 40 or 50%, which might translate to 2.5 liters or 2.5% predicted per year values. The question is really, what's the clinical meaningfulness of a treatment effect of, say, 2.5% on the percent predicted FVC, or say 3 or 3.5% on the percent predicted FVC. And again, just to point out, there's been a number of natural history studies that have shown this linear decline of percent predicted FVC and percent predicted peak expiratory flow rate in Duchenne patients. This is our CINRG data collected on over 300 patients, followed again for periods of up to 10 years, again showing this linear decline phase between 10 and 20 years of age. So the question in terms of assessing patient benefit of a treatment in non-ambulatory or advanced Duchenne patients, the question is, what does an individual patient identify as an important treatment effect? They may not experience an obvious change of, say, a 2.5% predicted value on a pulmonary spirometry test. They may not experience an appreciable difference of a 2.5% change relative to no intervention. So we oftentimes have to focus on doing some survey methodology and evaluating what's really clinically meaningful to a patient. There's also a lack of tools to measure quality of life or global clinical impression in Duchenne patients, specifically in those who have declining respiratory function. So this group, Santhera and academic colleagues, received input from the Community Advisory Board of the World Duchenne Organization. The Community Advisory Board recognizes the lack of specific tools measuring patient perspective in Duchenne patients with declining respiratory function. In terms of the, due to the lack of specific tools, the start of actual assisted ventilation, nocturnal mechanical ventilation, was seen as a major milestone influencing the life of a patient with Duchenne. And a delay in the need to start assisted ventilation was considered to be a patient-relevant goal by the Community Advisory Board. So this prompted us to ask the question, can we evaluate how slowing the rate of respiratory function decline influences the time to start of assisted ventilation? So just to show you an example here on peak expiratory flow rate percent predicted, we've also done similar analyses with percent predicted FVC, but using the CINRG natural history study data to do some modeling on this change in the rate of progression on peak expiratory flow percent predicted. So the objective, again, was to establish a link between the reduction in the annual rate of respiratory function decline and the time of starting assisted ventilation. So, for example, if the annual rate of respiratory decline was reduced from 5% to 3%, how much is the time from, say, a time of crossing 50% on the peak expiratory flow rate to the actual as a baseline parameter, so crossing of 50%. From that point in time, does this change in the rate of progression actually allow us to predict when assisted ventilation is going to be initiated? It does a slowing of the rate of decline actually lead to a prolongation of time to assisted ventilation. So the as an outcome variable, we use the percent, the peak expiratory flow rate percent predicted value as an example spirometry measure. We use time from crossing 50% of a PEF percent predicted value as a baseline to the start of any type of assisted ventilation as the clinical anchor. The 50% value of peak X-ray flow rate, percent predicted, was used baseline, as this is the typical average baseline of clinical trials conducted in non-ambulatory patients with more advanced disease progression in Duchenne. And I think we also acknowledge that there is a subjective nature to the decision to start assisted ventilation, and also systematic assessments of CO2 by either end-tidal CO2 or transcutaneous CO2 were not done in the CINRG study. And there's really lack of objective published data establishing a link of specific parameters of peak X-ray flow rate or forced vital capacity, CO2, and the actual start of ventilation in Duchenne dystrophy. With the new transcutaneous CO2 monitoring in the expanded Duchenne natural history study, we're actually hoping over the next 5 years to actually collect some really quite useful data that hopefully can have an impact on the clinical management of Duchenne in an evidence-based fashion. This is some of the data. On the left is the percent predicted FVC on the x-axis, and on the right is the percent predicted peak X-ray flow rate. And this shows the percentages of patients that are actually in the Duchenne natural history study, the CINRG study, that were free of mechanical ventilation within the ensuing 2 years as different measures of forced vital capacity, percent predicted, were reached, as well as peak X-ray flow rate. And as you can see, that crossing given thresholds of both peak X-ray flow rate and forced vital capacity does indeed correlate with a 2-year risk of assisted ventilation. But what the EMA wanted to see, again, was not just this relationship between peak X-ray flow rate and forced vital capacity and onset of future ventilation, because again, whatever threshold is chosen, whether it be a 50% value or a 60% value or 40% value, the thought was that was, again, a fairly arbitrary threshold that has been chosen by these expert panels. And really what the EMA was interested in seeing was are there actual forced vital capacity or PEF values that are actually associated with actual occurrence of CO2 retention and nocturnal hypoventilation as measured, for instance, by transcutaneous CO2 monitoring in the home environment. So the population for these analyses of onset of mechanical ventilation were extracted from the CINRG study. We extracted the relevant patient population, which was all patients who had crossed a peak X-ray flow rate of 50%, who had at least one PEF value above and below 50%. Both glucocorticoid users and non-users were included in the analyses. Patients from the site in India were excluded due to differences in standards of care. No other exclusions were made. And altogether, 103 patients were extracted from the data set. About 70% of them were using glucocorticoids. The mean age of these patients was 14.5. About 70% of these patients were non-ambulatory. And again, you can follow cohorts, for instance, groups of patients who declined less than the average. That would be the patients in the blue. So if the average percent predicted decline over the course of a year is about 5%, 5 to 6% in patients between the ages of 10 and 18, for instance. If you look at those who decline less than 5% per year, their average value is about 1.38% decline versus those that decline more than 5% per year. If you lump those into a second group, that is they have a 9.35% decline per year. And this gives a difference of about 7.95% is for as essentially a difference in the FVC percent predicted decline. And if you follow these two groups of patients over time, there's about a 3-year prolongation in time to ventilation in those with the slower or lower rate of decline versus those with the more rapid rate of decline in forced vital capacity. But again, this is a fairly arbitrary split of two groups of patients, just looking at those who decline less than the average and those that decline more than the average. So really, the question was, how can we split the CINRG data in other ways that will allow us to systematically assess how smaller differences in the rate of decline influence the time to ventilation. And so in this instance, we can't really split the groups to just above and below a given threshold, because this just does not generate sufficient small differences to assess the impact between these two groups of patients. So for instance, if you look at those you could look at those that decline 9% a year, those that decline 1% a year. But again, that's a pretty large difference in the rate of decline in forced vital capacity. Similarly, you could look at those that decline 8% per year and those that decline say 0% or have stable disease progression, and these are fairly large groups. So essentially what we need is overlapping groups in order to generate smaller differences. So for instance, if you take those that decline by 7% per year and all those patients that decline on average of 3% per year, there ends up being some overlap, so similar patients included in both those decline groups, but this allows us to then assess a smaller difference of, say, a mean difference of, say, 4% per year. So again, this is one concept, is the use of overlapping groups to generate smaller differences. So from a statistical point of view, the technique that was used was actually a statistical technique called bootstrapping. And we actually generated overlapping groups from these 103 CINRG patients and used statistical resampling methods. So this method called bootstrapping allows us to continuously allocate different patients into two groups for purposes of comparison. So for instance, you can have two groups with an average separation of 2% in the average rate of respiratory decline. So for instance, an average decline of 4% versus 6%. You can have groups with an average separation of 3% in the average rate of respiratory decline. So perhaps those with an average decline of 3.5% versus those with an average decline of 6.5%. And so we use similar procedures then to really assess all various degrees of separation. And for these separations, we perform Kaplan-Meier analyses to estimate how much of the separation in peak X-ray flow rate delays the time to start of ventilation. So this was the actual data generated from the bootstrap modeling, looking at the time from crossing a given threshold of 50% to time of actual initiation of assisted ventilation. So there in the upper curve, you can see that the differences there are resulted in prolongation of time to assisted ventilation from, say, 5 years to 6 years. There was other groups of separation which actually resulted in greater prolongation of time to assisted ventilation from, say, a 2-year prolongation from 5 years to 7 years, 5 years to 8 years, and prolongation all the way up to 4 years, from 5 years to 9 years. And then on the x-axis here is the actual separation in the annual rate of change in peak X-ray flow rate. So was it a 2% change or a 3% change or a 4% change in the two groups? And again, the y-axis is the actual hazard ratio for onset of mechanical ventilation. Again, there was important input from the Community Advisory Board of the World Duchenne Organization. Prior to conducting the analysis, we asked the CAB how long prolongation of ventilation-free time would be important. With input from the Community Advisory Board, the CAB suggested that prolongation of 1 year of time to mechanical ventilation would be of importance to a patient. So to give you an example of what the data actually showed here, here you can see the crossing of the threshold, a difference of 2.7% in peak X-ray flow rate percent predicted would result in prolongation of assisted ventilation by 1 year from say 5 years to 6 years. So 2.7% seems to be a clinically meaningful difference in the rate of progression. Here, a difference of 3.8% in peak X-ray flow rate results in prolongation of assisted ventilation by 2 years, from 5 years to 7 years. So in summary, again, based on the CINRG natural history data, a reduction of 2.7% in the annual rate of decline in peak X-ray flow rate leads to a 1 year prolongation of ventilatory free time after crossing this threshold of 50% on the peak X-ray flow rate. And because, again, a 1 year prolongation of ventilation-free time is important and clinically meaningful to a patient, a treatment difference then of 2.7% could be regarded as clinically meaningful on that measure of peak X-ray flow rate. We've done similar analyses for forced vital capacity percent predicted, which we'll share at an upcoming forum. So I'd like to just make some concluding thoughts, sort of vis-a-vis, I think, the importance of natural history data and the conduct of clinical trials in the era of the COVID-19 pandemic. I think it's certainly realistic that clinical trials can be conducted during a pandemic. We have to use a variety of strategies to be able to collect data. Video visits and other home-based strategies are key. Moving forward into new trials, we need to think creatively with regard to how data is collected in Duchenne trials. The COVID pandemic has created challenges for natural history studies. These studies need to be performed really in a quite consistent manner with current standards of care, as opposed to having patients come in for extra visits. The NSAA, the North Star Ambulatory Assessment, Time Function Test, PUL version 2.0, and pulmonary function tests have advantages as primary and as secondary outcome measures. We have a wealth of long-term natural history data. We know the prognostic relevance with regard to 1 year and 2 year changes using these endpoints. And we also have a wealth of data and have begun to understand the clinically meaningful treatment effects based on these specific endpoints. The new expanded Duchenne natural history study will create an additional test bed for the validation of home-based clinical assessments, such as NuvoAir, transcutaneous CO2 monitoring, video-based North Star, video-based PUL, and video collection of time function tests. So with that, I'd like to conclude and just acknowledge our great neuromuscular research team at UC Davis. I'd also like to acknowledge the support we've had from Parent Project Muscular Dystrophy. And I'd also like to acknowledge the many CINRG investigators worldwide, our sponsors of our natural history studies, as well as our industry partners. It's really taken a village to carry out this effort. We've received federal funding from National Institute of Disability Rehabilitation Research, NICHD, NIAMS, as well as Parent Project Muscular Dystrophy. and the Department of Defense, as well as NINDS. So again, I'd like to thank you for this opportunity, and hopefully would be available at some point to answer any questions you may have. Thank you very much.